Now, our new type of cell division in this unit is meiosis. So this is going to be a model of meiosis. And some people call this meiosis, and that's okay. Meiosis or meiosis is the same thing. Now, meiosis is kind of like all of the stages of mitosis twice. And I just have 6 post-it notes here. And so I'm not going to go through all of those stages, but we're going to hit the high points here. We start off with a cell that I'm going to call the mother cell. And in this mother cell, I'm not going to draw that mother cell in interphase, but I'm going to skip to prophase. And just like prophase that we saw in mitosis, prophase in meiosis has some similar hallmarks. And meiosis has 2 divisions. So I'm going to call this one prophase 1. Now, the nuclear membrane is dissolving, and inside the DNA has duplicated, so our chromosomes look like X's. There's big chromosome 1 and little chromosome 2 from the mom, and from the dad, big chromosome 1 and little chromosome 2. The DNA is packed up and ready to move, and the mitotic spindle is forming to help the DNA move to the right places. Now, at this point in the cell's life, the cell has 2 sets of chromosomes, a big one from mom, a big one from dad, a little one from mom, and a little one from dad. So we call this diploid, meaning 2 sets. Then, after prophase, we know that the DNA is going to get organized along the middle. And that's called metaphase, where M stands for the middle. And this is going to be metaphase 1 the first time. So let me put a new cell model down there to show our progression. And what's going to happen here is it's going to look a little bit different. Because what happens is the chromosomes line up in the buddy system, where the big chromosomes that are the same chromosome 1 line up together, the chromosome 2s line up together. They're organized this way by the mitotic spindle. So let's draw in that fully formed mitotic spindle there. And these chromosomes actually have a name to them, and they're called homologous pairs. And so in metaphase 1, the pairs line up. So that in anaphase 1, these pairs move away to either side. And then at telophase 1, so I'm going to write anaphase 1 because we're not going to use up another cell to show that. And telophase 1 is when these pairs are in 2 different piles at opposite sides. And then cytokinesis 1 is when this one cell is going to split into 2. So I'm going to tear this in half to represent cytokinesis 1 and this one cell splitting into 2. And now if you follow these along, you'll see what each cell has. So this cell on the left got the little green chromosome 2 and the big red chromosome 1. The cell on the right got the little red chromosome 2 and the big green chromosome 1. So now these cells only have one set of chromosomes. So these cells now are called haploid. And haploid means that they have one set of chromosomes. So this would now be considered prophase 2. So we skipped a couple of steps here, but what you can see is that the DNA is organized in a packed way, and now we'd start to get a mitotic spindle forming on either side. And that mitotic spindle is going to help organize these chromosomes along the middle of the cell. So let's take our next post-it. And I'm going to draw it like, I'm going to do this. Here we go. We'll just do it like that. And what's going to happen is this mitotic spindle is going to organize that DNA along the middle of the cell. So we've got our big chromosome 1 and little chromosome 2. Our big chromosome 1 and little chromosome 2. With the mitotic spindle. Same thing over here. So identical processes are happening in these daughter cells. Put that guy back over there. And then we've got anaphase 2. So this is metaphase 2. And before you saw that the pairs separate in anaphase 1. Well, in anaphase 2, what's going to happen is that these chromosome pairs, these chromosomes are going to actually split up, these chromatid pairs. So that's the chromatids split. So this part is going to move this way. These chromatids are going to move that way. And then we get telophase 2, where we have 2 equal piles of DNA within each cell. And then cytokinesis 2. Tears each of those cells in half. So what we've got is we've got this cell that was like this. Now that cell is being split in half again. And this cell is being split in half again over here. If we follow along, this cell would have half of a red chromosome 1, half of a double-stranded green chromosome 2, and this one would have the same thing. Over here, a big green piece and a little red piece. And now what we've got is cells that are still haploid. They've got one set. If we look at these cells compared to the beginning, they've only got half of the amount of DNA. And that's okay, because the cells that we've made here are cells that are called gametes. So gametes are sex cells, and the sex cells are sperm cells in males or egg cells in females. So you can imagine that each of these represents now a sperm cell. So sperm cells have this little part that's like a motor that's loaded with mitochondria and uses a lot of ATP And then the sperm cells have a tail called a flagellum. So we've got a flagella there. So what happens is meiosis creates all of these little sperm cells, or meiosis can create egg cells the same way. So this is meiosis. To recap, what we did is we started with a diploid cell. That diploid cell went through the process of prophase, metaphase, anaphase, and telophase, and split its homologous pairs into two different cells. At this point, we went from having two sets, big red, big green, little green, to only having one set, going from diploid to haploid. The next division split those sister chromatids into two different cells through prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2, leaving us with 4 different cells. These 4 different cells have different combinations of DNA compared to one another, and different combinations of DNA compared to our starting mother cell. That's the process of meiosis, creating gametes, sex cells, with different combinations of DNA compared to the starting mother cell.